The topic we will be discussing today is antigen-antibody interactions. But before diving into this topic, let's go through some concepts that you should be aware of first. We will go through: what is an antigen? How is it presented? What is an antibody? What is it like structurally? And what is meant by antigen-antibody interaction? Let's begin by talking about antigen. An antigen is any substance that causes your immune system to produce antibodies against it. It can be of different shapes and sizes. The presence of an antigen stimulates the B cells to produce more antibodies that can fight off the foreign invader. The three broad ways to define antigen include: an exogenous antigen is that one foreign to the host immune system. An antigen may be a substance from the environment, such as chemicals, bacteria, viruses, or pollen. An endogenous antigen is the one that is produced by intracellular bacteria and viruses replicating inside a host cell, and autoantigens are produced by the host. Normally, autoantigens would not elicit an immune response, but a sensitized immune system will mount an attack that results in the development of an autoimmune disease. A commonly known name for the presence of autoantigens is allergies. Now that we know that there are three types of antigens, let's move on and discuss its structural properties. It can be made up of peptides, proteins, and polysaccharides. A small region on the antigen determines its function and working. This small region is known as the epitope. It's the site that is recognized by the immune antibody. On the other hand, the part of the antibody that binds the antigen is known as the paratope. We will talk about it later when we discuss the structure of antibodies. Epitopes are further classified as conformational and linear epitopes. Conformational epitope. Conformation of epitope, or the 3D structure of epitope, defines its function. So in simpler words, the various shapes and structure of the epitope is recognized by the immune system and bounded by the paratope. Second is the linear epitope. It does not require any three-dimensional structure to determine its function. So, a linear or a sequential epitope is an epitope that is recognized by antibodies by its linear sequence of amino acids or primary structure. We have already discussed the three types of antigens, but an antigen can be further classified based on its function. These are the partial and complete antigens. The partial antigen is that which cannot elicit antibody formation, can elicit an immune response when coupled with a carrier protein. It is further divided into simple and complex hapten. The simple hapten is univalent and has a small molecular weight. On the other hand, a complex hapten has a large molecular weight and is polyvalent in nature, meaning it has two or more antibody binding sites. The complete antigen can induce antibody formation and an immune response without the help of a carrier protein. As for the antigen presentation and processing, we have already discussed that in detail in our previous videos, but just a quick review to help you revise some basics. The two types of antigens that are exogenous and endogenous antigens are presented by the major histocompatibility complex. The MHC contains over 100 genes which are organized into two major classes: the Class I MHC molecules and the Class II MHC molecules. Antigens are presented on the cell surface by the MHC1 and MHC2 protein classes, cytotoxic T cells, and MHC1 complexes. Antigens are presented on the cell surface by the MHC1 and MHC2 protein classes. A key feature of the MHC1 molecules is to recognize the healthy cell from the cell infected by some pathogen. You may think of it as a sort of quality testing. If a cell with normal peptides attaches to the MHC class I, it passes, but a cell with a viral peptide antigen is recognized and destroyed by the T cell. There is a mechanism behind this which we will get into now. Endogenous antigen produced within the cell is degraded in proteasomes, and antigenic peptides are transported to the RER, where they associate with the class I molecule being assembled. The class I molecule bearing the antigenic peptide is then transported to the cell surface and presents the antigenic peptide. Helper T cells and MHC EGA complexes. Unlike class I MHC, they are much more restricted in their expression. What happens is, exogenous antigen, which is produced outside the cell, is taken up by endocytosis. The endosome fuses with the lysosome, and the antigen is degraded. The endocytic vesicle delivers the antigenic peptides to a compartment for peptide loading, where the antigenic fragment displaces the invariant chain from class II MHC and becomes bound to the MHC molecules. The MHC molecules are then transported to the cell surface, where they present antigenic peptides. That is all about antigen. We will now talk about antibody in the next section.